Hello everyone, welcome to our channel Learn with Rajakumari. In this video, we are going to learn about Western blotting. Western blotting is specifically used for protein. In this video, we are going to learn about introduction, principle, procedure, advantages and disadvantages of Western blotting and applications. Introduction. Western blotting is a well-established analytical technique for detecting, analyzing, and quantifying proteins. This method is widely used to detect specific protein molecules in complex samples such as tissue homogenates and cell lysates. We all know that Western blotting is specifically used for protein hybridization. Western blotting is also named as protein immunoblotting because an antibody is used to specifically detect its antigen. That's why we are using the term immuno. Principle behind Western blotting. In Western blotting, target proteins, that is our protein of interest, are transferred to a hydrophobic membrane, that is nitrocellulose membrane, after sodium dodecyl sulfate polyacrylamide gel electrophoresis SDS page and detected using specific antibodies. SDS page is specifically used for the isolation of or electrophoresis of proteins. This picture depicts the cells and tissues are lysed to form cell homogenates, which is run on the SDS page electrophoresis. So here the proteins are separated based on the molecular weight. So then it will be transferred to the nitrocellulose membrane. After that, it will be probed with antibodies for the detection of target protein by an enzyme reaction. So this band gives the target protein. In this target protein itself, primary antibody is there. A secondary antibody is given in red color, which is already tagged with enzyme. So when we are adding this specific substrate which is suitable for the enzyme, it will form some colored chromogenic product. So it will produce some color. So based on that color, we can identify whether the specific protein is present in the sample or not. Steps involved in Western blot. 4 steps are mainly involved. Sample preparation, that is we need to get cell homogenate from the tissue. Second step, gel electrophoresis for the separation of proteins based on molecular size. Third step, membrane transfer. The band should be transferred to a solid support, that is nitrocellulose membrane. Fourth step, immunodetection, that is marking target protein using a proper primary and secondary antibody to visualize the presence. First step, sample preparation. Here we have to lyse the sample in appropriate lysis buffer, reduce and denature the sample with sample buffer containing sodium dodecyl sulfate, which is a detergent, and beta mercaptoethanol. Second step, gel electrophoresis. So after getting cell lysates, we need to do gel electrophoresis for the protein separation. So load an optimized quantity of sample onto the gel. Perform gel electrophoresis at 100 to 200 volt for 30 minutes and for 2 hours. Proteins will get separated according to its size, with smaller proteins migrating more quickly through the gel. Third step, membrane transfer. Prepare cold transfer buffer and the membrane. Assemble the transfer sandwich with the gel near the cathode and the membrane near the anode. The negatively charged proteins will migrate out of the gel onto the membrane. Perform this transfer at 30 volt at 4 degrees Celsius or 100 volt for 1 hour. Immunodetection. Use a total protein stain to check the transfer efficiency. Block the membrane in appropriate blocking buffer. Incubate the membrane with primary antibody 1 hour RT or 4 degrees Celsius. Incubate with secondary antibody 1 hour RT. Perform this detection step and perform data analysis. So in this picture depicts in the membrane the primary antibody is given in red pink color. The secondary antibody, which is already conjugated with horse radish peroxidase enzyme, is bound with the primary antibody. When the appropriate substrate, which is given in blue color, is added to the antibody, that horse radish peroxidase enzyme and substrate reaction will occur, which will give the colored product. So we can get the signal by colored signal. So this is the overall picture of Western blotting. So first uh, we have SDS treated protein mixture. So we need to add this sample to the SDS polyacrylamide gel electrophoresis. So we need to run the electrophoresis. So based on the molecular size, the migration of proteins will occur. Then remove the gel and perform electrotransfer. So we need to transfer the protein bands to the nitrocellulose membrane. Then bind antigen of interest with enzyme linked antibodies. So it will get bound with the protein of interest. So when we are adding the substrate, it will form colored reaction. So based on that color, we can able to understand whether the specific protein is present in the sample or not. So this is the basic technique of Western blotting. Advantages of Western blotting. Specificity and sensitivity is the main advantages of Western blotting. Disadvantages includes it is prone to false or subjective results, and also it has high cost and technical demand. Applications. Detecting different isoforms of proteins, detecting protein-protein interactions, detecting protein DNA interactions, then post-translational modification, then detecting protein subcellular localization, and also Western blotting used for the estimation of size of the protein as well as the amount of protein present in the sample mixture. Western blotting is also used as a confirmatory test for the diagnosis of HIV, neurocysticercosis, and tubercular meningitis. So I hope you understood about Western blotting. Thank you.